Bismillahirrahmanirrahim. 9700 November 2024, paper 1-1. This is an MCQ paper, which was the most recent exam that we had in November 2024. O Lord, give us in this world that which is good, and in the hereafter that which is good, and protect us from the punishment of the fire. As you all know, this paper is for 1 hour and 15 minutes, and please remember that the timings increased in the change in syllabus in 2022, and that's because the difficulty level has greatly increased in the MCQs. Micrometer, so it is actually 7 micrometers, so this will give you the magnification, which is 5000. Then coming to question number 2, which statement explain why lymphocytes with no nuclei die? No nuclei. So what is the function of nucleoli? The cells do not have mitochondria. There's nothing to do with mitochondria. The cells do not have mRNA, cannot transcribe, so it's not talking of transcription. The cells do not have ribosomes, because the nuclei are the site of ribosome synthesis, and so they cannot synthesize proteins. So that's why the answer is D. Question number 3, density gradient centrifugation is used to separate cell structures by their relative density. Larger cell structures have greater density and sink further down the centrifuge tube. So we know the nucleus is the largest, and then the chloroplast, and then the mitochondria, and then the ribosomes. That's why the answer to question 3 is D, because the smallest will settle down first, and then the big larger than that, and the large. So you've got to know which order it is. Nucleus is the largest, then chloroplast, then mitochondria, and then ribosomes. Now coming to question number 4, which cell structures contain nucleic acid? Nucleic acid means DNA and RNA. Now of course you have to remember that cell structures. So chloroplast, have a circular DNA, and then they have 70S ribosomes. So that's my 1 is correct. Golgi body does not contain any lysosome, does not contain. Ribosomes are made up of ribosomal RNA, which is a nucleic acid and protein. So it will be 1 and 4. That is why the answer to 4 is C. 1 and 4. So chloroplasts, you remember chloroplasts have a thylakoids, and then they have the circular DNA, and then they have 70S ribosomes. Now coming to question number 5, the statements describe processes that take place in a secretory cell. Modification of the protein occurs in the Golgi. mRNA leaves the nucleus, so this would be transcription. So first DNA to mRNA, then the mRNA goes out of the nucleus and then binds to the ribosome. So let's look now, read all these. Modification of the protein occurs in the Golgi body. mRNA leaves the nucleus. Ribosome bind to mRNA during translation. Transcription of a specific DNA sequence occurs. Vesicles fuse with the cell surface membrane. Vesicles transport the protein to the Golgi body. Now it says statement 4 is the first process, so we remove this one. And statement 5 is the last process. So what will come after 4? What is the correct sequence of the middle 4 processes? Right? Now the answer is B. Why is it easy? Well, I've just revised it with you because I'm sure all of you know that. So transcription of a specific DNA sequence occurs. That's fine. Then it will be mRNA leaves the nucleus. Fine. Then we have ribosomes bind to mRNA during translation. Then we have vesicles transport the protein to the Golgi. And then modification of protein in the Golgi. And then we know that it is the 5 was supposed to be the last one, because it says statement 5 is the last. So we know that after this there will be 5. After 1 there will be 5, and before this will be 4. So that is why the answer to this is B. Please revise protein synthesis if you're not very clear about it. Now coming to question number 6. How many types of structures with a double membrane that are found in animal cells are also found in plant cells? Now double membranes are where? Nucleus has a double membrane, mitochondria has a double membrane, and chloroplasts have a double membrane. Lysosome has a single membrane, and ribosomes do not have a membrane at all. Endoplasmic reticulum has a single membrane, Golgi body has a single membrane. So double membrane is only found, so it says those which are found only in animals. So nucleus will be found in plant cells, and mitochondria will be found in animal and plant cells. So that is why the answer to question number 6 is B, which is two structures, because you see chloroplast would not be present in animal cells. Then we come to question number 7. A naturally occurring polysaccharide synthesized in a plant is a branch chain of alpha glucose. The straight parts of the molecule are linked by 1,6 glycosidic bonds with only a small number of branches which are linked by either an alpha 1,3 or an alpha 1,4. Which polysaccharide has a structure most similar to that described? Now you know cellulose is beta, and you know glycogen is not found in plant cells. So you're left with a choice of amylose and amylopectin. Now the answer is branched is only amylopectin. Amylose is not branched. So it says the branch chain. So cellulose is beta, that was out. Glycogen is not found in plants, and that was out. Amylase is 1,4 only. Amylopectin is 1,4 and 1,6 and has branching as well. Now, question number 8, I think this was a rather difficult question. The diagram shows a triglyceride. An enzyme was used to digest this triglyceride into glycerol and fatty acids. Which scatter plot correctly represents each fatty acid component of the triglyceride? Now you can see there's a double bond here. So it says number of carbon-carbon double bonds. So this one does not have any. This has 0 double bond, this has 1 double bond, and this has 1, 2, 3 double bonds. So you've got to be looking at 0, 1, and 3. So because you see on the y-axis it has number of carbon-carbon double bonds and number of carbon atoms. So here you've got the number of carbon atoms. Now, why is the answer B? I want you to look at it very carefully. At 17, you can see these are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 carbon. So at 17 carbon there is 0 double bonds, and at 19 carbon there is 1 bond here. This is at 19 carbon because 17, 18, 19, we have 18, 19 here, and we have 1, and we also and the same, it's also because this is also 19 and this is also 19, and it has 3 bonds. So you have see the 3 here and the 1 here. I hope everybody is clear. Please do this paper before you see this video, because if you just listen to you will not really be challenging yourself to do these questions yourself. I will be posting an unsolved paper as well so that everybody can follow that, and first do the unsolved one and then you can follow this video. So for question number 8, the answer is B. As you can see, A, C, and D are all wrong, because you can easily figure it out from this answer, because there is in A, C, and D we've got 1 and 2 and then 4 double bonds as well. You can see here in this one there's a 4 as well, there's a 2 as well, and there's a 1 as well, and these are at 17 and 19, so these are wrong. Question number 9, the diagram shows how the alternating nature of beta glucose monomers along a chain allows hydrogen bonds to form between consecutive monomers. Please understand that, and then they've shown you the dotted line with the hydrogen bond. Now you can see the dotted line is here and the dotted line is here between two monomers. Hydrogen bond in cellulose affects the tensile strength, the ability to withstand pulling forces without breaking. How do the hydrogen bonds shown in the diagram help cellulose function as a suitable material for a cell wall? Now please try to understand when you read the answers is what was the question? How do the hydrogen bonds shown in the diagram? You've got to only refer to the diagram. A. The hydrogen bonds add additional tensile strength along individual cellulose molecules within the cell wall. Right? So this is correct. Along individual cellulose within the cell wall, because they're just showing you between the two monomers. Now, okay, why is B wrong? I want you to understand that. I remember somebody wrote a comment that means you do not explain the ones which are wrong. Because I assume that you all know your biology good enough, at least 70% to be doing these papers. The hydrogen, why is this wrong? The hydrogen bonds create stronger cross links between adjacent cellulose molecules. It is not talking about adjacent cellulose molecules. It says it says shown in the help cellulose function suited for hydrogen bonds shown in the diagram. In the diagram, we are only seeing hydrogen bonds between monomers. We are not showing between cellulose molecules. So this is wrong here. Stronger hydrogen bond between adjacent cellulose molecules. This is one cellulose molecule, just two monomers. Again, this is not in the diagram. Cellulose molecules within a cellulose microfibril. I mean, microfibril is many of these chains joined together and then forms a microfibril. Then you must know the structure of a microfibril. We're not seeing any microfibril. We're just seeing hydrogen bonds between the monomers in the diagram. It says shown in the diagram. So we're not talking of a microfibril. Microfibrils are made of many such molecules joined together. Question number 10. Two solutions 1 and 2 each contain a mix of two different biological molecules. One solution contains starch and sucrose and the other contains glucose and protein. Starch and sucrose. Starch will be positive with iodine. The two solutions were tested with a variety of reagents to identify the presence of the biological molecules in the solution. The table shows the results recorded. Which row identifies the two solutions? So one contains starch and sucrose. So the answer is C. Why is the answer C? Starch is present, so iodine is positive. In 2 it will be absent, yeah. In Benedict, so yes, it will be reducing sugar will be present, so starch and sucrose. Boil with Benedict solution. In 2 you have glucose, so 2 will have this positive for glucose here. And 1 will have sucrose here, so that with acid hydrolysis that will be positive. And why will it be again positive with acid hydrolysis? Because number 2 contained glucose and glucose would still show a reaction in 2 because it contained glucose, and protein would be positive in 2, which is glucose and protein. So that is why the answer to 10 is C. You have to understand is starch and sucrose, that means starch would be present, but then sucrose when it is hydrolyzed would only be present with this test, but would not be present with Benedict because sucrose does a non-reducing. You have to understand is glucose and all are reducing, but Benedict solution is reducing sugar test. But when you hydrolyze it, that's for a non-reducing sugar, which is sucrose. And biuret would only be positive with protein. Question number 11. The diagram shows parts of 3 pairs of amino acids within a protein. The pairs are labeled X, Y, and Z. So this is X, Y, and Z. And then it says which row shows the correct type of interaction that would occur between the two amino acids in each pair. Now the answer is B. Why? Because as you can see, this is an ionic bond. So I would have also concentrated on this. So this was it. So I would have fixed this one. Either this is right or this is right. Now Y, you can see, would have, what would be the ones you'd have to have hydrophobic interaction. And then hydrogen bond X would of course form a hydrogen bond between the two amino acids. Question number 12. A student carried out investigations at pH 1 to 8 to look at the effect of pH on enzyme catalyzed reaction. The optimum condition for this enzyme is the acidic environment of the stomach at pH 1 and 2, 1 to 2. The remaining substrate concentration was measured after 5 minutes. So if the reaction is occurring very fast, then there'll be less substrate available at the end. So remember the remaining substrate concentration. So initially we started off say 10,000 substrate, and were there now 500 or were there 400 left or were there 5,000 left? After 5 minutes at each different pH, which graph shows the effect of increasing pH on substrate concentration remaining after 5 minutes? Now we know that the optimum is 1 to 2. So at 1 to 2, there should be most least substrate. Now you can look at the four graphs. Now it makes sense. You see, the question is that which graph shows the effect of at 1 or 2 there should be least substrate. So in this there's a least. Now in this they're high. So this and this are totally wrong. I mean, students probably not reading it correctly to be writing an answer C or D. So what we need to understand is what would happen after some time when the pH is changed. If it is optimum was 1 and 2, after that there should be more substrate left. And then a time will come that, the substrate will become constant. It can't be decreasing after that. So this was wrong. And the only thing correct was A. Why? Because you see, as you increase the pH and the optimum is 1 or 2, as you increase the pH, the enzyme would not work. So the remaining substrate would be more. Question number 13. Which statement is correct for a non-competitive inhibitor? The inhibitor binds to the active site of the enzyme and decreases Vmax. That's wrong. The inhibitor binds away from the active site and increases the Michaelis-Menten constant. That's wrong. The inhibitor decreases. Yes, C is correct. The Michael does not change. The inhibitor does not change Vmax but increases. So this is wrong as well. Now I'm just going to show you a graph and you can probably understand this a little better now. As you can see, I've added this graph and you can see how the Km for A and B remains the same. Km for A and B remains the same. For C it changes, which is a competitive inhibitor. But the Km for A and B remains the same. This graph, of course, I've added to you. It's not in the question paper, but I've added for you to understand this so that you can understand it a little better. And I'll let you see it once again. Question number 14. Pyrophosphatase enzyme catalyzes a hydrolysis reaction. Pyrophosphate ion substrate, and this is the enzyme. It results in phosphate ion products. And pyrophosphate ion is the substrate. Now, experiment 1, a scientist studied the rate of this reaction using a colorimeter. The absorbance of the solution was measured at regular intervals until all the pyrophosphate has been converted into phosphate ion. So you can see the absorbance is increasing as the substrate is being used up. In experiment 2, the scientists repeated the procedure with a higher concentration of pyrophosphatase, that is more of the enzymes. All other variables were standardized. Please remember, pyrophosphatase is the enzyme. So in experiment 2, there were more active sites, more enzymes. Which graph shows the effect of increasing concentration of pyrophosphatase? Now, as we know that as the enzyme will increase, so the number of active sites will increase. So more enzymes means the reaction will be faster. Now, in all these situations, you can see the action because you see here the key is given. This is experiment 2, should have been faster. So experiment 2 is faster only in C. The rest of them is in here as well. But then you have to realize the absorption should become constant. It should become constant at a higher level. So this is wrong. So the answer to this is C. Question 15. Proteases are a group of enzymes that digest proteins. Which statements about proteases are correct? A bacterial protease that is secreted from a disease-causing bacteria could act as an antibody and cause the production of antigens. This is wrong. A bacterial protease secreted from the bacteria cannot be an antibody. Lymphocytes make antibodies. Then non-competitive inhibition of a protease that has an optimum signature pH 2 can be overcome by increasing the substrate concentration. Non-competitive cannot be increased by, it's only competitive inhibition in which can be increased. Number 3, water molecules are required when peptide bonds and link the monomers together are broken by the action of protease. Yes, this is correct. That's why the answer is D, which is 3 only. Then coming to question number 16, the diagram shows part of a cell surface membrane. Which row correctly identifies the molecules labeled 1 and 2? Now the answer was C. This is very basic, very easy. 1 was a glycoprotein and 2 was a phosphate head. This is a glycoprotein. If it was attached here, this will be a glycolipid. And this is the phosphate head, which is 2, which is the phosphate head. Now coming to question number 17, the diagram shows the mean dimensions of an epithelial cell in micrometers. So height is 3, length is 8, and width is 2. Which row shows the surface area to volume ratio of the epithelial cell and how the surface area to volume ratio of the cell would change if the cell. You see, if the cell becomes larger, of course, the surface area to volume ratio would decrease. So that is why the answer to 17 is C, 23 ratio 12, and it would decrease. Question number 18. Which diagram shows the correct direction of net water movement between the four cells due to osmosis? Now, what we need to understand is the answer is A. Why is the answer A? Because it always moves from a less negative to a more negative. So minus 22 to minus 40, less negative to more negative, minus 22 to minus 36, less negative to more negative, minus 22 to minus 33, and minus 36 to minus 40. So it will always move from a less negative to a more negative. I always make you remember it in a very easy manner. I says 22 solute, 40 solute. So this would have more free water molecules and this would have less free water molecules. Just imagine it. Minus 22 means 22 teaspoons of sugar in it, and minus 40 means 40 teaspoons of sugar in it. So that is why the question to 18 is A, the others are all wrong. You can look at it, they're going in the wrong direction. They've shown the arrows. It has to be from a less negative to a more negative. Question number 19. An investigation was carried out into the effect of 4 different treatments on the permeability of the cell membrane and tonoplast of beetroot cells. Beetroot cell vacuoles contain a red pigment. This pigment cannot diffuse through the tonoplast or the cell membrane. 1 centimeter cubes were cut from beetroot tissue and washed in running water for 20 minutes to remove any pigment released from damaged cells. 2 cubes were then placed in each of the 4 test tubes, containing different contents and observed for 5 minutes. Which row shows a correct explanation for the observation recorded for one of the treatments? Dilute HCl contents of the test tubes stay clear. Membrane proteins have been denatured. Well, then it should all become, then the pigment should leak out. B is correct. Why is it correct? Ethanol, contents of the test tube turned red. Lipids including membrane phospholipids have dissolved because lipids dissolve in ethanol. Water at 20 stays, membrane proteins have been denatured. Why would they denature at 20 degrees Celsius? Water at 80 contents test tube turned red. Lipids including membranes have dissolved. They cannot dissolve in water. Water at 80 degrees cannot dissolve the lipids. Lipids do not dissolve in water. Question number 20. The photomicrograph shows a stage of mitosis. And we can see it is metaphase. What would be correct for the next stage in mitosis? The metaphase, you know, as you remember, it's interphase, prophase, metaphase, anaphase, and telophase. So what would be the correct next stage in mitosis? 2 sister chromatids remain attached. No, they do not remain attached is the answer. Nuclear membrane is not present. It's not present in either prophase. It starts to disintegrate. Metaphase, no nuclear membrane. Anaphase, no nuclear membrane. In telophase, yes, nuclear membrane starts to reappear and then reforms. So the answer to 20 is A. We will stop this video here and continue it in the next video. Thank you very much.